Welcome to Conformational Analysis of Biotherapeutics by HDX-MS, presented by Genetic Engineering and Biotechnology News. Thanks for joining us today. I'm Tamlin Oliver, Managing Editor of GEN, and I'll be the moderator for this webinar. Hydrogen-deuterium exchange combined with mass spec is a powerful combination of technologies for the analysis of biotherapeutic protein conformation and examination of higher-order structural dynamics and epitope mapping. Advances in HDX-MS hardware and software not only offer scientists the ability to monitor changes in the higher-order structure of a therapeutic protein, but also to examine protein dynamics and association with other proteins and biomolecules. It's no surprise that its use is increasing rapidly. The four speakers today, St. John Skilton, Morten Beck-Trella, Stephen Berkowitz, and Hui Wei, have extensive experience working with and finessing HDX to meet their research needs. They will share their latest insights and enabling practices with us in just a minute. Before we get started, though, I want to encourage you to submit questions for our panelists at any time during the presentations. We'll answer as many as we can after the final presentation. You simply type your question into the Ask a Question box on the lower left of your screen and hit Submit. St. John Skilton, Senior Manager, Late-Stage Development, Pharmaceutical and Life Sciences Business Operations at Waters, is our first speaker. He'll give a brief overview on HDX Basics. St. John, we're ready when you are.
Hi, Tamlin. Thank you very much. Yes, it's quite fun being part of a technique that's becoming adopted by the mainstream, and it's really quite fun seeing some innovative science becoming part of reality. So I thought we'd start by looking at the sorts of terms that are used in HDX by LCMS. Next slide. There's a plethora of terms here that get used. Many of these are opaque to the non-expert, but in fact there are very sensible reasons for these terms. So one of them, as you'll hear from Hui Wei from BMS, is the difference between pulsed and continuous strategies. So pulsed, of course, looks at some kind of perturbation, some gross perturbation to see what happens when you do that, and the effect then on the uptake of deuterium, from which you can infer just how bad the change in conformation is, or how gross it is. And then continuous labeling looks at generally time courses, and this is the high-definition version where you can make dynamic comparisons. Next slide. We've illustrated here the difference between a global analysis of one conformation versus another, and really the information from this will be that conformation B will have a greater overall uptake relative to conformation A, and therefore you can say which one might have different characteristics in solution. So perhaps something is more open or more closed relative to another. Next slide. When you talk about local HDX-MS analysis, these are typically used for things like epitope maps and binding studies. And what's happening here, of course, is you're looking at bound versus unbound states. You enzymatically digest the protein or protein complex, and you end up with a direct comparison at a very local level, perhaps even in the best case scenario down to the amino acid residue, where you can tell exactly where the deuterium is, in relative terms, where the deuterium has been taken up by one peptide versus another. So that gives you a very solid map of what exactly is happening, and it's perhaps the most challenging in terms of the overall data mountain that you end up with. Next slide. It's clear that the use and actual studies involving HDX are increasing dramatically, and this was highlighted back in 2009 by John Engen, who drew up a graph of all of the citations and publications, and you can see year on year how it's been increasing steadily, and of course it's reached even further up in the last couple of years. And the studies are extremely varied. Next slide. And of course, a lot of these studies have demonstrated some key aspects. Firstly, the utility. This can be summarized here, the fact that hydrogen-deuterium exchange by mass spectrometry is being used in real-life situations in industry, and it's no longer the preserve of academia, and this will be highlighted by Steve Berkowitz. And then also the applicability of what's being studied is a great deal higher, so you're starting to get down to very small differences in conformation that are looked at that have a great overall effect on efficacy. And that's a real-world problem, and you need to know exactly why these changes might occur. And it's something, of course, that regulatory agencies are taking a very close look at and actually starting to see data being submitted to them by people using HDX. And then very importantly for us is the usability. So we, of course, have to make tools that people can use on an everyday basis. And this involves both software and hardware. And what we're trying to do, of course, is make these things as automated as possible and as usable for the non-expert who might be a biologist to then be able to use that information and use that in a real-life setting much more rapidly than was the case in the past. So you'll see that again in all three of the speakers that are going to follow this. So for us, it's a very exciting time. We can see all of these applications and these studies going in at very high levels and getting very high rates of information out. And we're very happy to be a part of that. So back to you, Tamlin, and I hope you enjoy the rest of the speakers.
Thank you, St-John. Our next speaker is Morten Bech-Trella, a postdoctoral fellow in Thomas J.D. Jorgensen's research lab at the University of Southern Denmark. Morten will talk about his use of HDX to assess the local flexibility of active PAI-1. Dr. Jorgensen, his advisor, will join us for the Q&A later, so feel free to address questions to Morten, Dr. Jorgensen, or St-John at any time. Morten, you're up.
Thank you, Tamlin. And first of all, I would like to thank Waters for giving me the opportunity to present some of our data here today obtained by hydrogen deuterium exchange mass spectrometry on a protein RNA complex. The title of my talk is Effect of RNA Aptamer Binding on the Structural Dynamics of Plasminogen Activator Inhibitor 1. And this work was done in collaboration with Peter Andreasen and his team from Aarhus University in Denmark. Next. Plasminogen Activator Inhibitor 1 is a serine protease inhibitor, or Serpin. It's the major inhibitor of plasminogen activations in the humans. It regulates fibrin degradation, and this is most clearly seen by the fact that elevated PAI-1 levels is associated with increased risk of thrombosis. Decreased PAI-1 levels are associated with a mild bleeding tendency, and furthermore, high PAI-1 levels are associated with poor prognosis for survival in a number of human cancers. And that actually makes PAI-1 not only a prognostic marker, cancer marker, but also a therapeutic target. Next. This slide shows you the inhibitory activity, or the mechanism behind the inhibitory activity of serpins in general and also of PAI-1. And to the far left, you see the structure of a mutant of active PAI-1. You cannot crystallize the wild type currently. The molecule is characterized by the protrusion of a reactive center loop, shown in orange, and by the yellow arrow. And to this loop, the protease binds, and that is shown in the middle picture. The protease is colored purple. And that binds to the loop, cleaves it, and this cleavage triggers translocation of the protease to the other pole of the molecule. And that transition is actually driven by the insertion of the reactive center loop into the central beta sheet A, which is colored red here. And the complex to the far right illustrates this, and this is not of PAI-1. We don't have a crystal structure of PAI-1 with its target protease, so this is of another serpin. Next. PAI-1 is a little bit special because it has the ability to spontaneously insert its reactive center loop into the central beta sheet A, and that is what is illustrated here on this slide. And it does so at physiological conditions in the order of 1 to 2 hours. And the result of that reaction is that PAI-1 is inactivated, and this structure is called the latent form of PAI-1. Next. So we have all these crystal structures that very nicely visualizes the endpoint of these structural rearrangements of the molecule, but we don't really know in very much detail what the underlying mechanisms of rearrangements are. And of course, we would like to investigate this a bit further, and we are particularly interested in the latency transition of PAI-1. So one major question is, why does PAI-1 convert to the latent form when other serpins don't? And in order to get closer to an answer to this question, we think it's important to investigate the structural flexibility of PAI-1, and we have used hydrogen deuterium exchange to do so. And in this presentation, I will show you some studies on hydrogen deuterium exchange on PAI-1 without any ligand bound, but also with these RNA aptamers, which are actually inhibitors of the latency transition. Next. So here's a slide that just depicts the HD experiment that we have been conducting. And to the far left, we start out with the protein in water, and we then initiate the exchange reaction by dilution in the D2O. We let that go on for various time intervals, and we then quench the reaction by lowering the pH to 2.5 and the temperature to 0 degrees. The labeled protein is then digested with pepsin, and the generated peptides are separated by liquid chromatography, and the deuterium content of the peptides is determined by mass spectrometry. Next. And here is a picture of such an instrumental setup. On the left, we have my boss standing in front of one of our older instruments where you have basically just a normal HPLC system, and then you have some homemade valve and tubings which can be submerged into an ice bath in order to keep the temperature low during the separation of the peptides. And while these systems work really well, the ice bath does give some inconvenience, especially when you try to find a leak in the system. A few years back, it was determined to try to develop a better solution and came up with this HDX unit, which can be integrated into the nanoacuity UPLC system. And it's basically just a cooled chamber wherein you have all the valves and tubings that you need to keep cooled. And we've been part of the prototype test program of this unit, and I must say that we are really pleased with the added convenience of this instrument. Next. Here is shown what it looks like when you have acquired all your data of all your deuterated peptides. So at the left, we have on the bottom there the undeuteriated peptide, and then you can follow the increase in mass as the peptide takes up deuterium. And on the top, we have a full deuteration control, which is generated by labeling in the presence of 8 molar guanidinium chloride. And we can then calculate the deuterium content and plot it against the exchange time and achieve a graph like the one on the right. And we can also plot the full deuteration control, and that's the dashed blue line there. Next. And if we then look at all the different deuterium uptake plots for all the peptides, we get something like this, which obviously contains a high degree of quantitative information about the deuterium uptake throughout the molecule, but it's a little bit difficult to interpret this data in relation to the three-dimensional structure of the protein. So what we really like to do next is to create heat maps of the data. So we take the deuterium content plots and we calculate for every peptide the relative deuterium content. And we then map that along the amino acid sequence. So we have the N-terminal of pi1 shown here, and you have in the gray boxes below the amino acid sequence the identified peptides, and beneath that we have the relative deuterium content, the relative to the full deuteration control, in beneath gray boxes there for each time point measured. And we then use a color scale as indicated, and what you can then quickly get an overview of is which peptides are highly flexible, that's the dark red one, and which one are more protected against exchange, that's the white ones. And you can also reference this information directly to the structural elements mapped top of amino acid sequence. Next. And if we then look at the entire heat map of pi1, it looks something like this. And we find that these heat maps are absolutely excellent reference charts when comparing many different states of experimental states that you want to compare in your study. But although beautiful, they're not very well suited for presentations because of the information content. So what we would rather do is actually somehow transfer this information to the three-dimensional structure. And that is what I've tried to do on the next slide. So next, please. And what you have here is the 3D structure again of pi1. And I've grouped the peptides into 3 categories. White are those with relative deuterium contents less than 60%, and the light red is 60 to 90%, and above 90%, which is basically very close to full exchange, are colored dark red. And if we then start out here at 0.1 minutes, you can see that peptides in the lower right part of the molecule are beginning to take up sufficient deuterium to get above the 60% line, but in the rest of the molecule we're still below 60% deuteration. Next, please. If we then move on to the 1 minute time point, a lot has happened. Suddenly many peptides are now above the 60% cutoff throughout the structure, and even some peptides in the lower right corner are already close to fully exchanged. And also what you can see is that, and indicated here, is that the core beta sheet B, indicated by the circle, is actually acquiring deuterium at a slower rate. Next, please. If we then look at the 10-minute and 100-minute time points, we see this trend continuing. More peptides in the lower right corner get fully exchanged, while the central or the core beta sheet B remains relatively unexchanged. So what this shows is that you actually have a molecule where the flexibility is polarized in the sense that the lower half of the molecule is way more flexible than the top half, and especially than the core beta sheet B. Now that we have achieved the basic understanding of the local flexibility, the next step was obviously to move on and investigate the changes in local flexibility when we inhibit the latency transition using our RNA aptamers. Next. These RNA aptamers against Pi1 was developed by Peter Andreasen, our collaborator from Aarhus University in Denmark, and his team a few years back, and that was published in Biochemistry in 2010. They developed two RNA aptamers named PioNAP5 and PioNAP40. Both bind active form of pi with low nanomolar affinity, and they also both delay latency transition. PioNAP5 delays the latency transition by 4-fold, and PioNAP40 delays it by 8-fold. Importantly, none of them actually affected the inhibitory activity of the Serpin. Peter Andreasen also went on and tried to identify the interaction surface between the aptamer and Pi1 by site-directed mutagenesis, and that data is displayed on the structure in the middle of the slide. And they were able to identify a bunch of residues colored red here, which were important for binding of both aptamers, and they also identified one residue, that's the purple one, which was important for binding of PioNAP40. Next. So what we did now was that we repeated the experiment and just added in 2-fold molar excess of the aptamer before the deuteration was started. Next. And before moving on to the full results, I just want to show you a couple of examples of peptides that were protected by the aptamers. And this one is from the peptide colored blue on the structure there on the right. And this peptide contains two of the residues that were found important for binding of both aptamers. And actually what you can see is that both this peptide is highly protected by both aptamers. Next. This is another peptide, and this peptide contains one residue that was important for binding of both aptamers and one residue that was only important for binding of PioNAP40. And again, you can see that both aptamers induce protection in the peptide, but PioNAP40 induces a slightly stronger protection compared to PioNAP5, which correlates nicely with the fact that this peptide contains two residues that were important for binding of PioNAP40, whereas only one which was important for binding of PioNAP5. Next. So now we look at the entire heat map again in order to get an idea about the global effect of PioNAP5 binding to Pi1. And I start out again here with the heat map of PioNAP1 without the aptamer bound. And then we add in, right beneath there, a heat map. So next. Of the same thing, but now with PioNAP5 bound. And what you can see in the top row is that you have some peptides that turn from dark red all the way to white, indicating a strong protection in that region. Next. And we then add in a third block and that in this block we calculate the difference between the two upper ones and color that according to white to blue color scale, so that nearly white indicates peptides where there's no difference between pi without PioNAP bound and with PioNAP bound, and the dark blue ones are those that gets highly protected by PioNAP5 binding. Again, this all looks very beautiful, but it's a little bit difficult to interpret the data in terms of the three-dimensional structure. So I, again, have tried to transfer this information onto the three-dimensional structure. So that is shown on the next slide. And what you see here is that actually peptides all over the lower half of the molecule gets highly protected by PioNAP5. Not only peptides in the region where the sites, the residues that were important for binding of the aptamers were protected. So these data indicate that PINUP5 induces strong protection in more or less the entire bottom half of the molecule. Next. We can then move a step further and compare this difference map to the relative deuterium content map that I showed you previously. And what is striking here is actually that those peptides that was previously found to be the most flexible ones when no ligand was bound is also the ones that gets most protected when we bind these aptamers. And actually you get a similar result for both PINUP5 and PINUP40. The question is now to what extent this dramatic stabilization is also caused the reduced rate of latency transition. Based on the data shown here, it is very plausible that the dramatic stabilization of the lower half of the molecule would work to slow down the rearrangement to the latent conformation. Although our data does not directly answer the question of how and why Pi1 converts to the latent state, it provides a very informative overview of the local flexibility of Pi1, and that this flexibility is modulated upon aptamer binding. Next, please. Finally, I would like to summarize by saying that we have used the HDX to assess the local flexibility of active Pi1, and we find high protection against exchange in the core beta sheet B Pi1. We find low protection against exchange in the most of the lower half of the molecule, and when we bind PINUP5 and 40 to active Pi1, we see strong protection against exchange in the proposed binding epitope on Pi1, and we see increased protection against exchange in almost the entire lower half of Pi1. Finally, PINUP5 and PINUP40 stabilizes many of the otherwise most flexible regions of the protein. Next, please. Finally, I would like to thank a few people. First of all, Thomas Jørgensen, who is my boss, Daniel Hirschberg, who's old postdoc in Thomas's lab, who started out analyzing Pi1, Peter Andreasen from Aarhus University, and two of people, Jeppe and Daniel, did the development of the RNA aptamers and the biochemical characterization. Mikael Plow from the Finsen laboratory in Copenhagen was also part of this project since the start of it. And finally, I would like to thank the Danish Council for Independent Research, Technology and Production Sciences for funding of the project. Thank you very much, and back to you, Tamlin.
Thanks, Morten. If you have any questions for Morten or any of our other speakers so far, please submit them now. Our third speaker is Stephen Berkowitz, Principal Investigator, Analytical Development at Biogen IDIC. Stephen will speak about his strategies to mine valuable information from HDX-MS data. Stephen, ready when you are.
Thanks, Tamlin, for the introduction. And today I'm going to be talking to you about a simple and practical scheme for presenting all the HDX mass spec data that one can actually acquire from these experiments. And I'm hoping that previous speakers have laid the foundation for you to understand what is actually involved in doing these experiments. And we'll be extracting higher information from protein structure of these biopharmaceuticals. But one of the problems is actually looking at all this data, and that can be a problem. Today, though, we'll be looking at interferon as a model for this. Next slide. So this is interferon, and this is a molecule we'll be looking at in terms of trying to explain the data analysis and presentation. And what we'll be doing is looking at some of these residues being modified, the methionines or the free sulfhydryl, and then comparing that with the unmodified. So we'll actually be doing some comparability experiments, and these comparability experiments are actually very important in the biopharmaceutical industry, and you can see that deuterium exchange mass spectrometry could play an important role in providing higher order structure information about these molecules in terms of comparability. Next slide. So, hoping from the previous talks that have gone on before, we have an idea of what's actually involved in a deuterium exchange mass spectrometry experiment. And what we have here is the amino acid sequence of interferon and the various peptides that we have actually obtained from the pepsin digestion process. And again, this is a local deuterium exchange experiment, also referred to as a bottom-up type of experiment in terms of proteomic nomenclature, you might say. So here we have the peptides lined up in terms of their sequence, and we have about 94% coverage. So we can actually look at almost the entire molecule with this technique. In this case, we have about 67 unique peptides that are common to the reference material relative to the experimental samples that we're going to compare to that reference material. In this case, interferon is actually a pretty small molecule. You get into antibodies, you wind up getting hundreds of peptides, and that's a lot of data and a lot of information that we need to process. But one of the things you notice that there seems to be a lot of overlap, which you might think is a sort of a waste of information, but that actually turns out to be, can be very important for redundancy purposes of accuracy of what you're getting in data. Also, it's capable of being used to provide higher resolution information. Hopefully, in the near future, using electron transfer dissociation process, these peptides can be further digested or cleaved, I should say, and we can actually approach one amino acid resolution, but that's in the near future. Next slide. So what we want to do is, starting this experiment, is we have 67 peptides, and we start off at 0 time with the mass of these peptides known. We then basically incubate the protein in deuterium exchange for various periods of time. In this case, throughout this discussion, we were actually going to take 5 data points. Next slide. And what we're going to do is basically look at the mass increase, these delta M's, for the reference material peptide and the corresponding experimental peptides, and we'll be generating these mass differences. So we're really going to look at the relative mass change in each of these peptides over time. Next slide. In so doing, every peptide that we have identified will have a corresponding data set of 5 time points, and therefore 5 different mass increases during the course of the experiment, and then be 1 set for the reference peptide and 1 set for the experimental. Next slide. So that we can take each of those peptides, looking at the experimental and reference, and make for each peptide a plot, as indicated here, of the deuterium exchange in uptake. As you can see, there's lots of data to actually process, and as I mentioned, with something like an antibody, we'd have over 100 or several hundred of these plots, and that can be sort of overwhelming. Next slide. So what we're trying to do to simplify this process is we go back and we line up these peptides again relative to their amino acid sequence position in the protein, as indicated at the top. So those peptides are aligned. Next slide. We then locate the midpoints of each of those peptides and then display them on the bottom x-axis. And what we have now is each peptide is equally spaced along the x-axis in the order determined by each peptide's midpoint in the protein's linear sequence relative to the amino terminus. So the I value for each of those peptides is determined by those midpoints, and each of those midpoints are equally spaced along the x-axis. And now that x-axis is actually a quasi-equivalent representation of the protein. It's not a direct one-to-one amino acid positioning of those peptides. So that's why we were calling it a quasi-sequence representation. But to give you some perspective in terms of when we plot this data, in terms of the linear sequence of the protein, what's actually going on. But again, once ETD, electron transfer dissociation, is developed, this will be much more approaching a one-to-one correspondence. Right now, we just have a quasi-representation at the bottom on the x-axis. Next slide. So now what we want to do is we have these mass changes for each peptide, and we want to convert those to fractional, and that turns to be rather more informative to basically get an idea of what's going on, since some of these peptides are short, some of them are long, and without normalizing that, we really get a good perspective of what's actually going on in terms of whether it's entirely exchanged or very little exchanged. So what we do is divide each of these mass difference data points for each peptide at each time by the maximum number of potential hydrogens that could exchange, and that number is unique to each peptide and is determined by the number of amino acids minus the number of prolines plus 1 for the one amino terminus. So that's how we can actually convert this data now into relative fractional exchange. Next slide. So now we're going to begin to sort of take all this data, and we'll start with one plot. We have this plot of fractional exchange for a given peptide over as a function of time, and we're looking at the reference in black versus the experimental in red. And so we plot that data, and this is the plot we got. And as we mentioned, there's lots of these plots. So what we're going to do is take one plot, next slide, and then basically take the experimental and just simply multiply it by -1. So we've more or less got a mirror image plot where we flip the experimental to the bottom Next slide. We then take each of these data points for the experimental and the reference and transpose it over to the y-axis at x equals 0. Next slide. And we then simply move all those data points over to its correct I position for that peptide. And we do this for all the peptides. In so doing, we get a plot that looks as shown here. Next slide. And what we have might look like a butterfly plot or a mirror plot, and it gives you a good relative sort of comparison, in this case, of interferon, which is made at the top through normal procedures, whereas the bottom is interferon made in a different culture media. In this kind of scenario, you would need to do a comparability study to show to the agency, if you're submitting this for drug approval or for IND approval, that, in fact, the molecule hasn't changed. And looking at this data, you can see, have the next slide. We can see that, in fact, qualitatively, it doesn't seem to show any difference. And again, remembering that, as indicated here in this little rectangle in the plot, we're looking at these deuterium exchange plots for a given peptide. So we can easily see that in some cases the peptides are changing over time, whereas others, they don't change at all. In some cases, they change, but after they very rapidly change, we see no further change in the deuterium exchange uptake. So by making this plot, we get an overall general picture of what's going on with various parts of the molecule in terms of the exchange, plus we have an ability to make this relative comparison between the experimental and reference peptides. And in addition, all the data that we've acquired during this experiment is actually presented in this plot. And this is actually very helpful to take advantage of all the data that's acquired. Okay, next slide. So now what we want to do is take it to the next level and actually do a quantitative difference assessment. And what we're going to do is for each peptide and for each time point, do the mass difference for the reference and the mass difference for the experimental at each peptide, each time point, we're going to take that difference and get difference number. And so that way for every data point now, we can calculate a difference number between the experiment and the reference. And we can also accumulate for each peptide, we can sum up all the time points and look at the difference in terms of a single parameter. So the sum of the difference. So that's specific to the entire peptide. So we have these two different numbers that we can actually calculate. And one thing to keep in mind, the way we have it set up here, is that if we have positive numbers, that means the reference is exchanging more. And if we have negative numbers, the experimental peptides or data sample is actually exchanging more. And next slide. This basically shows several very common exchange plots, and it basically indicates that you typically have, as indicated, like an A, C, and D, very little difference, just a random noise between the experiment and the reference, and that would indicate that there's really no change. And then there's other scenarios like B, E, F, and G, where there's a constant change that progresses. So summing these numbers just reinforces themselves, whereas when you have no difference, such as A, C, and D, by summing the numbers, you basically get a very small number because there's a cancellation of the pluses and minuses. Where you're going to run into some problems, and this corresponds to that second type of difference number. Next slide. At H, where the actual crossing occurs, and there the difference where we sum the differences for each peptide time point could give us a little problem, and we'll have some alternative ways to deal with that. So this is generally the kind of scheme where we're taking advantage of these different types of deuterium exchange uptake plots in terms of generating these difference data. Have the next slide. So now what we're going to do is plot the individual difference data for points as indicated here, and then we can connect the common time point in terms of a particular color. So now you can actually see for all the peptides at, say, 240 minutes, the black line corresponds to all the peptides at exchange at that time point, how much change in mass. Now, next slide. Now, what we can also do is plot some of the total exchange. In other words, each data point for a peptide, each time point is summed, and we get the total sum of difference as indicated at the bottom here. And instead of putting these as individual data points, we're just going to actually plot them as bar plots. And by doing that, we can actually then combine both these data points, as shown in the next slide. so that we have the individual data points as little points connected by different color lines, each line color corresponding to a particular time point, and then the bar indicating the total sum of differences for that peptide. So you can see obviously data can be positive, can be negative. Next slide. Next couple of slides are actually going to deal with how to deal with the accuracy of this data, because what we want to do is develop some kind of confidence limit so that we can assess whether there's a difference or not in these comparisons. So if we looked at the data points in any given experiment that we were conducting in terms of a comparison, we usually do the experiment at least 3 different times. So for each data point, for each peptide and each time point, we actually can calculate an average and therefore a standard deviation. So if we look at that average standard deviation as a function of the size of the peptide, as indicated at the top, we don't see any bias. It seems to be independent, roughly speaking, of the size of the peptide. If you look at in terms of the average, any given time point, again, we don't see any indication of a bias associated with time. And if we look at the actual amount of deuterium exchange on the bottom, you can again see that there seems to be very little indication of any bias. So what we've done is, with these standard deviations that we've calculated for these data points, which can in this case be over 1,000, we just simply take the average. Next slide. So that average actually comes out to be about 0.14 Daltons. So that represents what we feel is sort of an unbiased estimate of the standard deviation. And having that number, we can then actually calculate a confidence limit using the equation just below, which is basically nothing more than using a student t-test for a given confidence limit and number of degrees of freedom, which is equal to the number of experiments minus 1 times the standard error of the mean. So that's a little simple equation that gives us the capability of calculating a confidence limit. And in this case, next slide, we have 98% confidence limit. When we have 3 experiments for any given data point, we have an uncertainty of about plus or minus 0.5 Daltons. In the case of the sum of the differences over the time points for each peptide, that uncertainty can be calculated by simple propagation error analysis, and that comes out to be about plus or minus 1.1 Daltons. Next slide. So we need to then develop a quick criteria, and using these important confidence limits, what we have here at the top, if no data point for a peptide exceeds 0.5 Daltons, and then that peptide, in terms of the experiment, are the same. Even if the sum exceeds 1.1, it's critical that we at least have this 0.5 exceeded. On the other hand, if one or more data points exceed 0.5 limit and the sum of the differences exceeds the 1.1 plus or minus, the reference and the experimental peptides are considered different. We can get into a little complication. Again, this is because of the crossover in that earlier plot, the deuterium exchange plot, that can occur by simply summing the differences. In that case, if in fact we do have the individual data point exceeding 0.5, but the sum don't exceed 1.1, then what we do, instead of summing the individual differences, we instead sum the absolute values. This way we can deal with the crossover. And now if the sum exceeds 1.1, we consider the reference and experiment different, and if it doesn't, then they're considered the same. So this is a criteria that we've generally used to assess differences for a given peptide from the deuterium exchange data we acquire. Next slide. We can also actually calculate difference parameters by summing, in this case, for each peptide, using the sum of the differences for each of the time points, subtracting 1.1, and then summing those over all 67, we can come up with an index. But if this parameter inside each of these terms is below 0, because we can potentially get negative numbers, we'll assign a 0 there. We're only dealing with positive numbers. And if the sum does not exceed 1.5, we'll give the entire index a value of 0. Next slide. For each individual point, instead of dealing with each peptide, we can actually sum for every data point that we acquire, do a difference between that number and the 0.5. And again, I should mention earlier that we need to take the absolute values of these, again, because some of them are negative. If we can sum all those data points and come up with a second index. Again, if each of these terms, though, inside is below 0.0, we'll assign a value of 0. So then we need to sum the data, and if the sum does not exceed 0.55, we'll assign 0 to that index. So we have a way of now calculating these indices. So the next slide. It shows a plot of the final difference plot where we have the individual data points connected by colored lines, plus we have the bar plots, the sum of the individual time points for each peptide, and then we have the confidence limit indicated for both these difference numbers, and then we have the index. So we have a simple plot now that gives us a quantitative assessment of the difference. Next slide. So now we're going back to this interferon comparison where we grew the interferon in our standard culture and compared it to our experimental. So we have this mirror plot, butterfly plot, which we've seen earlier, but on the bottom we now have the actual quantitative difference plot. And as we can see, we cannot see any difference at all, as indicated also by these indices on the right-hand corner. Next slide. So this is a situation where we're comparing interferon, where we'd actually have a reference at the top, and we're comparing an interferon molecule that's been alkylated, and we can simply have a very controlled reaction at that free sulfhydryl. So that's the only position in the entire sequence that's modified. You can see right away in those two squares at the top that there's actually some differences that show up right away qualitatively, whereas the bottom plot is the quantitative difference plot, and we can see very striking differences, changes in the molecule that have occurred over a fair amount of the region of the sequence. And again, we have very significant numbers to go with our indices to indicate quantitative level of change. The next slide. This is a comparison where we're looking at interferon reference versus oxidized interferon. In this case, we treated the interferon with hydrogen peroxide, and in so doing, we've modified the methionines, and we also modified that free sulfhydryl. And again, looking at the top at the butterfly plot, we can very readily see differences qualitatively between these two molecules. And then on the bottom, we can then see quantitatively in terms of differences. Again, very striking differences in the structure along the sequence. And again, the indices, again, indicating even greater change in terms of differences. Next slide. And this is the last sort of example I have here. Is what we're going to be doing is looking at factor 9 and factor 9 FC. Factor 9 FC is a fusion protein that Biotherapeutics actually is in the process of trying to develop. And if we look at the top plot, what we're looking at is a commercial factor 9. That's a commercial product that exists. And this molecule binds calcium, and calcium is very important for its function. So what we're looking at is a comparison of commercial factor 9 with and without calcium. You can see there's very striking changes in the molecule along its sequence in terms of this difference plot. And the bottom is the actual F factor 9 region of the factor 9 FC molecule, the fusion protein. Again, we're looking at the comparison of with and without calcium, and you can see actually qualitatively the pattern of change is very similar, and that's what we would hope. All we're doing is fusing two molecules together, the factor 9 with the FC, and we're hoping that there's no further changes in that molecule. And this data qualitatively seems to indicate that's actually the case. But if we now do a direct head-to-head comparison, on the right-hand side at the top, we have factor 9 versus the factor 9 region of the fusion protein with calcium. And again, we make that comparison, we see no significant differences, whereas at the bottom, we make that comparison with calcium. Again, we see no significant difference. So again, pointing to the fact that when these two parts are fused together to make this fusion protein, that in fact we have little impact on the conformation of the factor 9 molecule. Next slide. So that concludes this presentation. Let me just thank finally Damien Hood, who's actually worked with me and has actually done all these experiments. I want to thank Damien very much. He's done a really great job, and we want to thank our collaborators, John Engen, Northeastern University, the staff at Waters, and Dr. Igor Kaltashov at UMass Amherst, who we've collaborated with, and finally our head of our department, Rowan Matra, Biogenidec. So let me now turn the presentations back over to Tamerlan.
Thanks, Stephen. If you have any questions for Stephen or any of our other presenters, you can submit them now. We have one final presentation, and that's Hoi Wei, Research Investigator 2 at Bristol-Myers Squibb. Hoi will provide two case studies, one involving global HDX-MS and a therapeutic mAb, and the other focused on local HDX-MS and GCSF. Hui, we're ready for you to begin.
Thank you, Tamlin, for the very nice introduction, and good morning, everyone. Today I'll talk about two case studies to show you how in biopharmaceutical industries that we used the HDX-MS technology for some comparison studies of protein therapeutics. Next slide. As the previous speakers have introduced, there are many unique advantages in HDX-MS technology. HDX only requires a very small quantity of protein sample compared to other techniques. With local HDX-MS, the localized information in conformation can be obtained, whereas the other methods, such as CD, cannot. So there are many potential applications for this technology, and they are listed on this slide, such as to study whether a protein has folded correctly or not, or whether certain processes make the same protein or not. For example, in the batch-to-batch comparison, or site-to-site comparison, or even the comparison between innovator versus biosimilar. And we can also use HDX-MS to study if there's any conformational changes after certain treatments, such as in different formulations and during the stability testing. And we can also use it to see if certain mutation along the protein sequence would affect the protein structure or not. It can also be used to identify binding sites between antibody versus target proteins, and this is a very popular topic now, which is also called the epitope mapping. Similarly, it can be used to identify binding sites of drug with target proteins, and it can also have the potential to identify the sites of protein aggregation. Next slide. The first case study I'll show you is to use global-level HDX-MS to do a comparison study of therapeutic monoclonal antibodies. As you all know, monoclonal antibodies are of great therapeutic significance. The ability to quickly and effectively characterize these proteins is of great importance, as it will help to facilitate their transition through discovery into development, eventual commercialization. Therefore, we need a series of appropriate analytical tools to provide an array of chemical and physical information during all stages of discovery and development. In this case study, we characterized a conformation of a recombinant therapeutic monoclonal antibody using global HDX-MS. We evaluated the reproducibility of the global HDX-MS and used the method for comparison studies of a monoclonal antibody in different formulation conditions. Next slide. So this is the system set up for global HDX-MS in our lab. It consists of an QD UPLC to deliver fast desalting of the proteins, an ice box that keeps the columns and most of the tubings at 0 degree, and a TOF instrument that accurately monitors the change in protein mass in the HDX experiment. Next slide. Quickly, for an overview of the workflow for global HDX, we look at exchange level of intact proteins to see how much deuterium uptake occurs for the whole protein. So quickly, we incubate the protein in neutral buffer at room temperature, followed by diluting the protein in 20-fold of excess of deuterated buffer, such as D2O. Let the protein labeling occur for a different amount of time, and then quench the reaction by reducing the pH to 2.5 and reducing the temperature to 0 degree. And then we do a fast online desalting on UPLC, followed by the high-resolution MS to measure the amount of duration incorporation. Next slide. We first evaluate the reproducibility of the method. I did the 3 full-time course HDX experiments within the same day and in 3 different days. The figures on the left are the relative deuterium uptake curves for the intra- and inter-day reproducibility study using the same monoclonal antibody. The tables on the right show the numbers for the average deuterium uptake level and the percentage CV at each time point. As you can see, as the labeling time point increased, the amount of duration also increased, and the percentage CV at each time point was less than 10% in both inter- and intra-day experiments. Overall, only micrograms of sample were used, so the instrument is of high sensitivity and the method is of a high reproducibility. Next slide. We then compared 2 samples using global HDX-MS. There are 2 conditions. Condition 2 is the final product, and condition 1 is the one step away from the final product. The difference between the two samples was with or without 0.05% polysorbate. Comparing the two samples at each labeling time point, the p-values were greater than 0.05. This means that there is no significant difference observed for HDX at all time points. Along with other biophysical data, this data is part of the submission document for regulatory filings. Next slide. The second case study I'm going to show you is how we use the local HDX-MS to analyze granulocyte colony stimulating factor, or GCSF. GCSF is the most potent inducer of terminal differentiation to granulocytes and macrophages of leukemic myeloid cell lines. It consists of 175 amino acids, and the pegylated version of this protein has a 20 kilodalton PEG attached to the N-terminus. So why compare pegylation? Because many final protein therapeutics are pegylated for better stability and drug delivery. There have been changes in protein structure observed for many pegylated proteins. It is challenging to study pegylated protein at intact level due to the heterogeneity of the PEG for many techniques. So it will be interesting to use peptide-level HDX-MS to reveal more detailed structural differences between non-pegylated and pegylated protein. Next slide. This is the system set up for the peptide-level HDX-MS. The picture on the left shows the Waters NanoQD HDX system with the LEAP auto-sampler. With this setup, we can perform HDX experiments with full automation. We use a 9-mobility QTOF instrument, which is SYNAPT G2 for peptide identification and HDX measurements. We use Waters PLGS and Dynamics software for peptide identification and processing the HDX data. Next slide. HDX-MS combined with pepsin digestion is capable of identifying individual regions along the protein sequence undergoing exchange. This is the workflow for the local or peptide level HDX-MS. It is very similar to the global level HDX, other than that at the LC step, instead of using a desalting column for intact protein, a pepsin digestion column is used for online digestion of protein into peptides, followed by the nano-QT-LC reverse phase separation. In order to obtain a localized comparison between the pegylated and non-pegylated GCSF in the HDX level along the entire sequence, it is important to identify common peptides that can be produced from both proteins in each experiment. For this purpose, 3 control experiments were performed on each protein to obtain the common peptides. In the control experiments, H2O instead of D2O was used for the labeling step to facilitate the peptide identification. Next slide. Then after the database of the commonly identified peptides was obtained, the D2O labeling starts to determine the deuterium uptake level for each peptide of the protein of interest. Next slide. A total of 6 online digestion control experiments for both proteins were performed. The control data were processed in PLGS software for pepsin peptide identification. 46 peptides were unambiguously identified in both proteins in at least 4 out of 6 runs, constituting 91% of the peptide backbone of GCSF with overlapping peptides in multiple regions. The residues that were involved in receptor binding are highlighted in the sequence. Secondary structure elements such as alpha helices and loops are also indicated in the sequence. The high sequence coverage allowed us to monitor and compare deuterium incorporation in local regions between the 2 proteins, revealing conformational differences and subsequent impact on the protein's biological activity if the changes are observed in or near the receptor binding sites. Next slide. The 46 common peptides were then monitored for their deuterium incorporation level in the labeling experiment. The chromatograms on the left show the 12-minute fast separation of peptides under 0 degrees nano-QD UPLC. The peptides are well separated. That allowed us to monitor the deuterium exchange level of each peptide. On the right, these are the spectra of the peptides of the peptide that elute around 5.6 minutes at each labeling experiment. As you can see, the mass of the peptide gets higher and higher as the labeling time increased. And this trend is similar between GCSF and pegylated GCSF. The mass of each peptide from both proteins at each labeling time point were recorded, and their relative deuterium incorporation level were calculated. Next slide. With the data, we can plot the deuterium uptake curves for all of the common peptides for both proteins for the entire map. The blue curves are the relative deuterium uptake level for GCSF, and the pink curves are the relative deuterium uptake level for pegylated GCSF. The y-axis shows the maximum number of amide hydrogens that can be exchanged for each peptide. Each one shows various uptake amount and different exchange rates. Some curves show no change between control and pegylated, and some curves show difference. And these plots continue on the next slide. Next slide. Here we chose to show some representative peptides that did and did not have different deuterium uptake, and to locate them in the three-dimensional X-ray crystal structure of GCSF, which was obtained from the literature. For the peptides with the curves shown at the top, they're the ones that did show a difference between the two proteins. For example, peptide 5 to 12, this peptide locates very next to the pegylation sites, and the pegylated version of the peptide has a quicker exchange rate than the unpegylated version. Another peptide, which is 115 to 122, it is also located closer to the pegylation site in the 3D structure. It also showed more rapid exchange rate in the pegylated version than the non-pegylated version. The peptide 83 to 89, it is a peptide in an alpha helix region, and this one is also have a higher deuterium exchange rate in the pegylated version than the unpegylated version. But many more peptides did not show a significant difference between the two proteins, and many of them are the peptides in the loop regions, such as peptide 93 to 104 and 125 to 138. There's a peptide which is 19 to 32. It is a long peptide in the alpha helix region, and this peptide has multiple residues that are involved in the receptor binding. And this peptide, we did not observe any difference between the two proteins. And the result indicate that although we do observe obvious difference between the two samples, however, these differences may not have an impact on the protein's biologic activity. Next slide. A quick and simple way to visualize the comparison is to plot a butterfly chart that compare the deuterium incorporation levels for all common peptides between GCSF and pegylated GCSF in multiple labeling time points. This plotting scheme was very recently introduced by Dr. Ho and co-workers and was also introduced in the previous presentation. Briefly, the chart is a mirror plot with GCSF on the top as a reference and the pegylated GCSF on the bottom. The y-axis is the relative fractional exchange, and the x-axis is the peptide position ranked by the midpoint of the peptide in the protein sequence. The different colored curves represent the relative deuterium level at each individual time point. For this plot, one can easily compare the relative deuterium incorporation at peptide level across the entire polypeptide backbone with spatial and temporal information. Next slide. For a more quantitative comparison, a difference chart was created from the difference of the deuterium uptake between the two proteins as shown in this slide. The x-axis is the same as the one in the butterfly chart, and the y-axis represents the difference in the HDX level in delta between the two proteins. Each vertical bar in the chart represents the summed difference of all time course for a corresponding peptide. When the value of each bar is above the black dotted line, which is plus minus 1.5 Dalton, then we consider it as significant difference in deuterium incorporation comparison. In addition, it is also considered a significant difference at individual labeling time point when the individual difference value on the curve is greater than 0.5 Dalton, which is represented by the blue dotted line. You can see that there are multiple regions along the protein sequence that show the significant differences in HDX level between the two proteins. But when we take a closer look, these regions do not include the binding regions. Therefore, the biological activity of the protein may not be affected by pegylation. Next slide. Finally, we evaluated the intraday reproducibility of the method by drawing the difference charts using the replicates from the same proteins. The top plot is the GCSF, and the bottom chart is for the pegylated GCSF. As you can see, the curves and bars are well confined within the significant bands on the plots, indicating the good reproducibility of the methods. It also told us that the differences that we did observe between the two proteins were real. Next slide. In conclusion, HDX-MS plays an important role in the characterization of higher-order structures of biologics. In a global level HDX-MS, it is highly reproducible, very rapid, and can be automated for high-throughput therapeutic protein characterization. For the local HDX-MS or pipette-level HDX-MS, is also very reproducible and with much improved throughput with automation. It can reveal very small local protein differences induced by pegylation. Both global and local HDX-MS have great potential in biopharmaceutical industries for applications such as comparison analysis and many more. Next slide. Finally, I would like to give my acknowledgments to these people. Firstly, I'd like to thank these brilliant scientists from Waters Corporation for software and hardware support, especially Jumi An, who had significantly contributed her time and effort into the work on the GCSF. I would also like to thank Professor Engen at Northeastern University and Dr. Hode at Biogen IDEC for helpful discussions. I would also like to thank my colleagues at Bristol Myers Squibb for their selfless support. And thank you all for listening, and back to you, Tammy.
Thanks, Hui. Before we begin the Q&A session, I want to encourage you one more time to submit questions for St. John, Morton, Stephen, and Hui. Also, Morton's advisor, Dr. Jorgensen, will join us for the Q&A, so feel free to throw him questions as well. We're ready to start our Q&A discussion. We have a lot of good questions, and we'll answer as many as we can. Our first question is from Morton. Morton, which software was used to determine the extent of HDX exchange?
The software we used was the Dynamics software that we also mentioned from Waters, and that was used to extract all the mass spectra from the raw data and generate the deuterium uptake plots, and the data was then transferred into Excel, and the heat maps were then made in Excel.
Okay. Hui, how much does ion mobility help?
Yes, ion mobility is a separation of ions in gas phase, within milliseconds of time frame, and the separation is on the fly during the LC-MS runs. So although the separation is good in UPLC, however, it is within a few minutes of radiant under 0 degrees, so we do see a lot of overlapping peptides. With ion mobility, it adds another dimension of separation, and which allows us to further separate these overlapping peptides. So this is especially useful when you have large proteins, such as monoclonal antibodies or protein complexes. So in general, you gain about maybe 20% of more peptides using ion mobility versus without ion mobility. So this helps to improve the sequence coverage and also improve the spatial resolution of the HDX experiment because you will have more overlapping peptides in the sequence.
Okay, thank you. Stephen, how much has the automated software cut down the time of analysis?
Well, that very significantly. I think if you go back a few years, you would take on the order of maybe a month or two to process a lot of this data. The advances that have occurred up to now, it can bring it, well within a week's time and even faster.
Okay, St. John, we have a bunch of questions for you right now. The first one is, what do the FDA and European and Japanese regulatory authorities think of HDX?
Well, I can't necessarily tell you what they think because that's sometimes a private conversation between themselves and the people who submit it. But we do know that the regulatory authorities have received HDX data and have looked through it. So it's a reality now. Don't forget the regulators are very careful to say that it's up to the submitter to explain why they're doing it and what they interpret from it. But what I must point out, of course, is that we've created these tools in response to what industry's asked us, and industry, of course, is in constant communication with the regulators. So the tools that they've asked us to develop reflect effectively what needs to be submitted from that point of view.
Okay, when will the commercial system be available, and is Waters the only one?
It's available now. Yes, we're the only ones to supply a commercially available system to our customers. It's already, in fact, had 3 years of field experience through an alpha and a beta program, so it's a fully working and fully operational system. And it's the sort of thing that, again, people have asked us to make, and we've gone and made tools in response to what they want.
Okay, and what's the purpose of using the cold box?
The reason we developed that is because it's one of the ways in which you can cope with back exchange. So back exchange is the reversal of the deuterium uptake, and you want to try and control that so you can sort of lock in the picture of what you've got to take that snapshot. And the ways in which it's done in solution would be typically through pH regime, so you quench through pH, and by keeping it cold, you also manage that back exchange very effectively to try and minimize it or get rid of it completely if you can.
Okay, St. John, I have another regulatory question. You suggest that this data can be submitted to regulatory agencies. Does this mean the software and methodologies used to produce and analyze the data are fully GLP compliant?
Well, let's take a step back from that. We started off with this tool as a characterization tool. So at any stage, people have obviously been submitting HDX-MS data to the regulators as part of supplementary information and supporting information to their overall filings. As far as the informatics itself goes, today all of the data and the reports can be captured through SDMS packages or LIMS systems, and that meets the compliance, the contemporaneous archiving factor for that sort of world. But going forward, so looking into the future, we're going to incorporate obviously the workflow for things like release testing into our Unify platform in the near future. So that means it'll be fully compliant ready at that stage.
Okay. Dr. Jorgensen, I have a question for you. How does HDX protection at the flexible region compare to the X-TAL structure B factor?
That's a very interesting question, and so the correlation between the B factor and the protection factor has been investigated by Walter Englander in a paper from '98 in Protein Science. And from protection factors ranging from 1,000 and up to millions, then there is no correlation between B factors and protection factors. So that is the level of protection. But for regions that are very dynamic, where the protection factor is very low, then I would expect a correlation, because then there would also be some static disorder in the crystal that would be reflected also as very fast exchanging amides in solution. So again, for very flexible regions, I would expect a correlation, but as soon as the protection factor exceeds 1,000, then there's no correlation.
Okay, thank you. Steve, I have some questions for you right now. The first one is, could you explain why the difference chart is so useful, especially as it uses the midpoint of the peptide?
Yes, I think there's a lot of comparability studies I think I mentioned that are very important in the biopharmaceutical industry. That's something that's done quite frequently with a lot of companies, and things that can bring more sort of quantitative assessment of that comparability as opposed to a qualitative assessment, saying two spectra or two different types of thermograms are the same. I think anything that can bring it to a quantitative level brings that comparability into a more rigorous state, and I think that's important. The concept of the midpoint is a way of locating the peptides and putting them in order. It doesn't translate into actual difference, but yeah, that's my take on the difference quantitative aspect.
All right. Are we expecting to see the use of HDX by biosimilar manufacturers?
I think that's going to be. I really think that's going to be a reality as things progress. This technique offers a level of comparison that you'd have to go to something like X-ray diffraction or NMR, and there's obviously problems with those types of techniques from a biophysical standpoint. So I think deuterium exchange mass spectrometry gives an opportunity to really look at the overall structure at a high coverage and actually make that comparison to assess the comparability in terms of the higher order structure.
Okay, the next question, Steve, I'm going to have you answer first, and then Hoya, I'm going to ask you to comment on as well. Can the local mode of HDX by LC-MS pinpoint the actual location of conformational change?
Well, there's a potential of reaching, I guess, a maximum limit of one amino acid, but by and large, at the present time, the resolution on an average basis is somewhere. I think that there's some discussion about where that is, but it probably varies. But something on the order of 3 to 6 or 7 or maybe even 10 amino acids is sort of your typical resolution. But in some cases, you could bring it down to the maximum. But once we input something like electron transfer, electron dissociation into the process, that could bring down to that one amino acid resolution.
Okay. Hoy, do you want to weigh in on that question?
Yeah, I completely agree with Steve. So usually you get about a few amino acids of spatial resolution in the pipette level HDX, and with the assist of MS-MS, like the electronic transfer dissociation, or even with the assist of I-mobility MS, you help to improve that spatial resolution as good as maybe single amino acid. So yes, that's exactly what the local level HDX can do.
Thank you. Steve, another question. Why do HDX studies use so many time points?
Well, I think the realization is there's a lot of dynamic properties that are going on in these molecules, and there are people who do single point measurements to get data, but clearly there's kinetic processes related to the exchange of the various amide protons, and you need a wide range of data to cover that, and that's all important information. So the more data you can take, the longer the range, the more unique information, the greater the possibility of it making a more critical assessment. So it's basically that you're dealing with a number of different entities exchanging, and how well you can characterize that exchange, how many data points you need.
Okay, Stephen, I have one more question for you. How do these plots vary when multiple data sets are analyzed, and are there any additional statistical insights?
Well, I mean, that's, we think this is an important aspect in trying to assess, comparability, is try to bring this to a more quantitative level. And so far, we've taken some very elemental steps to bring that to a very quantitative level. The data that we report are averages, and we do at least 3 measurements over several different days. And we've come up with these concepts of confidence limits, and those confidence limits are expressed at 98% confidence levels. So the points that are on those graphs are averages, and we define those limits. If they're exceeded, then that there's actually some kind of difference. So in my presentation, I did try to present a number of approaches how we assess those quantitative confidence limits. They're very elementary, but at this point, this is our best take at trying to bring a level of statistical uncertainty into these measurements and then making that conclusion.
Okay, Hoy, we have time for one more question, and I'll direct it at you. What is the experimental variability in terms of DA? So to claim the DA difference we observe is real?
That's a good and very practical question. So based on my experience, for individual labeling time points, I see usually around 0.1 to 0.2 Dalton of reproducibility. And if you want to give a confidence band of, say, 98%, you multiply that number by 3, so you are giving 0.5 to 0.6 Dalton of confidence band. And for the summed value of all the time course, I see usually about 0.5 Dalton, and so that gives us about 1.5 Dalton of the confidence band.
Okay, thanks, Hoy. Our time's up. Thanks for listening in, and thanks for all the great questions. I'm sorry we couldn't get to them all. This webinar will be archived for 6 months on our website, www.genengnews.com. If you missed parts of it, you can watch it again, or you can recommend it to friends and colleagues. Once again, I want to thank St-John, Morton, Dr. Jorgensen, Stephen, and Hoy, as well as our sponsor, Waters Corporation. We'll be sending you a survey shortly and hope you'll take time to respond. Your comments help us to continue to provide topical and timely webinars. Thank you very much.